So thank you for the invitation to speak with you today and introduce the NIH Comparative Genomics Resource project we have happening at NCBI and the National Library of Medicine at NIH. My name is Valerie Schneider, and I'm the lead for this project. I want to begin by thanking many of the people that have been involved in the work I'll be telling you about today. These include NCBI product owners and their development teams, NCBI and NLM leadership, as well as various committees that provide project oversight. And before I dig in, I want to let you know about additional ways in which you can learn more about recent project updates and outreach events for CGR. These include our recently updated website, the CGR mailing list, and following us on social media. I'll provide these links again at the end of my presentation. The CGR project aims to maximize the biomedical impact of eukaryotic research organisms and their genomic data. The CGR mission is to establish an ecosystem to facilitate reliable comparative genomics analyses for all eukaryotic organisms through an interoperative suite of NCBI repositories and knowledge bases offering high-value data, tools, and interfaces compatible with community-provided organism resources. While users will still be able to access NCBI data, tools, and interfaces as they always have, CGR will provide an additional access point to connect with tools and resources important to comparative genomics research. The goals of the CGR project are to promote high-quality genomic data submission with cloud-ready public tools, to facilitate comparative biological analyses by providing seamless, streamlined access to fair genomic data for all sequenced eukaryotic research organisms, to enrich NLM-held genomic-related content with community-supplied sample metadata and gene functional information, and to support workforce development, especially in the context of building a cloud-conversant data science workforce. Development decisions for CGR are being informed through community engagements. With those intended goals in mind, I share this cartoon, which provides a more detailed view of the CGR ecosystem to introduce the anticipated organization of content and tools. In the core, NCBI is responsible for foundational genomics-related content and some organism-agnostic tools. The content includes genomes, genes, transcripts, proteins, taxonomy information, literature, as well as gene orthologs for all eukaryotic research organisms. Communities at large support other types of content and resources and may also build tools that operate on NCBI core content to support cross-organism or organism-specific analyses. These include organism-specific knowledge bases, variant annotations, image collections, as well as genotype-phenotype relationships. NCBI will also provide standardized infrastructure and tools that enable the community tie-in of the overlapping circles. I'll now briefly share updates on a few of the current CGR project activities. High-quality genomic data is foundational to reliable analyses. Today, we find that more than half of the eukaryotic genome submissions to NCBI are contaminated with sequence that isn't from the intended organism. And if the contamination isn't removed, it can lead to errors in the analyses. And if the contamination isn't discovered until the submission of the genome, the requisite cleaning of the data can dramatically slow its public release. So to better address these issues, we wanted to improve the existing GenBank screening process outlined here and make it available as a public tool, meeting the goals on the left-hand side of the slide. We now have public beta releases for two parts of this pipeline, shown in red, as containerized tools available on GitHub. FCS Adapter identifies and trims contamination from sequencing adapters, a very common source of contamination. And FCS GX is a brand new cross-species aligner tool that identifies contaminants from non-source organisms. Terrence Murphy introduced the underlying methodology for this tool at last year's biodiversity meeting. GX runs 2 to 3 orders of magnitude faster than the current NCBI process with substantially improved sensitivity and specificity. We've also started using FCS GX to improve the data currently in GenBank and RefSeq and have already removed more than 130 megabases of contamination in collaboration with the original data submitters. A CGR-associated analysis tool is clustered NR. NCBI created this new BLAST database in order to provide reduced results redundancy and faster searches. Derived from the default BLAST protein NR database, by clustering sequences at 90% length and identity, it lets users see hits across a wider range of organisms and evolutionary distances. This image here shows the Choose Search Set section of the Protein BLAST submission form where you can find this database. And to help users assess it, we offer the option to perform simultaneous searches against the clustered and the standard NR. We've also built new web interfaces to help users find and retrieve genome data that have been informed by extensive user testing. In the search bar at the top of any NCBI page, users can now just type in an organism name and get a straightforward knowledge panel that connects them to relevant data and tools. Clicking on the organism's name takes you to a new organism page. And on the organism page, you can browse all genomes and find information for the reference assembly, annotation, as well as genes. I'll give a quick shout out here to our newly redesigned genome assembly web pages, which include assembly statistics, sample details, as well as information on assembly methods, annotations, and revision history. They also offer links to related resources like PubMed Bioproject and other genome-related tools. Returning now to the species page, you can also investigate the availability of genome data at different taxonomic levels by clicking this Browse Taxonomy button. The Download button provides fast access to the download of a user configurable package with sequences, annotation features, and metadata reports for the reference assembly. Users can also create download packages for other genome assemblies and for genes. For those who want programmatic access to the genome and gene data, we also offer command line tools and a RESTful API. Check out the presentation by Nuala O'Leary at the same meeting to learn more about the powerful programmatic interfaces to these data. We also have a new web tool, the Comparative Genome Viewer, released in beta just a few weeks ago, which allows users to compare two genomes based on assembly alignments provided by NCBI, making it easy to see structural changes. We invite you to try it out and give us feedback, particularly about the organisms and alignments it includes. And to learn more about it, check out the poster at this meeting by Sanjita Rangwala. Structural annotations of genomes are a key component of comparative analysis. However, 80% of the eukaryotic genomes submitted to GenBank lack structural annotation, and of the 20% that have submitter-supplied annotation, more than half have annotations in which more than half of the proteins are named as hypothetical, drastically limiting their use for comparative analysis. We're also beginning work to transform the RefSeq eukaryotic genome annotation pipeline, or EGAP as it's known, into a public cloud-ready containerized workflow. And in doing so, users will be able to run high-quality annotations independently before submission. This activity is aimed at getting better data into GenBank. We're also planning to expand the pipeline to support the structural annotation of small eukaryotes such as fungi and protists. In the past year, we've additionally been putting a lot of work into the curation of superfamily architectures on proteins to lay the groundwork in support of protein naming and functional annotation. These architectures are now indexed and publicly available on several protein resources at NCBI, including the conserved domain search results, Sparkle architecture summaries, and the protein family models database. Annotation will be a long-term effort in the CGR project, an area where we would like to engage communities for their input. Other activities that we're working on, but I don't have time to talk about today, include efforts to expand support for orthologs and emerging pan-genome representations. So if you're interested in learning more about CGR, I encourage you to check out our webpage and follow us on social media. And if you're interested in opportunities that are upcoming to give feedback on the CGR effort, I urge you to sign up for our mailing list. I want to thank you again for joining today and in advance for contributing your thoughts as we prioritize development features and connections for the CGR project.